Now, obviously, you appreciate that in some of those applications, you're going to think about polyepsilon caprolactone biodegradable polymers. So the degradable CVD coating is going to be very handy in this context as well. Here we're going to nanostructures or to smaller structures using AFM. So we're basically doing dip pen nanolithography on those surfaces, again with a click type reaction to immobilize proteins. This is material independent. Here we show it for different types of polymers. Here PMMA, for instance, stainless steel, and even like a glass surface. Now, the coating itself is superior to a spin coating in the sense that it's conformal and can take on three-dimensional or can coat three-dimensional objects very nicely. And to prove that case, we took just a bunch of glass beads, basically, coated them with a particular polymer. This is a polymer that has a benzophenol side group, so it's a photoreactive side group here. So this reacts with light, and then used 3D, projection lithography to pattern onto those glass beads to show the effect of three-dimensional coating. And so this works quite nicely. Here's a human hair that was modified in this particular case, but it also works with microfluidic systems, with stands, for instance, which are three-dimensional, but also really with 100 micrometer glass beads, for instance. So the limitation in resolution here is really more coming from the projection lithography, which has become much better now than before. And so I think we could even go to micron or maybe even submicron, maybe not, but micron level patterns with those systems as well. So here is like a kind of an extension of that work where initially we modify a flat polymer film. So this is like a polymer substrate that has the form of a film. We pattern that, and then we basically combine this with thermal forming so that's kind of the process that is used, for instance, to make your yogurt beakers, to then create local well structures. So you can make essentially micro well structures with intrinsic patterns already built into them. In this case, we're using two different types of CVD polymers. One is a ATRP initiator, so that is subsequently modified or initiates essentially the formation of a polyethylene glycol type material, a PECMA, which then gives us protein resistancy. So the backbone is protein resistant, so cells don't like to stick to that. And then here we immobilize through this activated ester group. We immobilize adhesion molecules to then guide cells into those patches. And then in conjunction with the thermoforming, we can create well structures that really allow us not only to place cells in there, but also then locally pattern within those well structures, which is something that is really difficult to do otherwise. So here's the confirmation of those data, and you see the cells are actually really following those patterns as well here on the bottom. Okay, so that's kind of a little bit of a background. This is certainly a topic of interest in my group for a while. And so then a few years ago, a colleague, Nick Appert, at the time he was in Wisconsin, I was in Cornell, approached me and said, well, we have done a lot of work with liquid crystalline materials. Couldn't we just basically take a thin cover of the CVD coating, put it on a droplet of LC material, and then could we use that as a sensor material? Because as molecules bind to the functional groups, you should see a deformation in the LC, which should be giving you an optical signal. So that was a very simple case. Okay, no problem, we'll try that. Unfortunately, the data were very inconsistent. And so when we started to look into this a little bit more, in particular, Kenneth Chang, like a graduate from the material science department here in Michigan, he started to look into that a little bit more in detail. And what he found is, well, actually, it's looking very different. So rather than getting a film on top of the LC, the monomers are actually diffusing into the polymer, starting to polymerize on the bottom, and then basically are being templated into nanofibers. That was interesting because now we have essentially this ability to topologically control surfaces as well. So instead of getting only thin films that are conformally coating the surface, we can now essentially also create this well-defined arrays of nanofibers. And so here you see a few different polymers that we use. Again, this alkyne, we like that a lot because we can modify it easily. Copolymers, I didn't talk much about this, but we can make copolymers with this too by just combining power cyclophanes and then the base polymer here. Now the thickness of this polymers is very well controlled. So we show you two examples here. And so that's another silent aspect of this work, that we can, through CVD, control the thickness very nicely. So that's important here as well, because as we control the thickness, we can then basically understand the growth kinetics of the nanofibers in those LC phases. And so let me just skip up this a little bit. So from a mechanistic study, what we learn is when the LC itself is in a crystalline solid form, then we don't get the formation of nanofibers. So we have an anisotropy in the system, but we lack the fluidity for the system. If we go to a isotropic phase, then on the right side, then we have the fluidity, but we're lacking the long-range order. And so in the absence of that, we also don't get nanofibers. Only if both the fluidity and the anisotropy are combined in form of a nematic liquid crystalline phase, we get the formation of nanofibers. And we have basically shown this here systematically. So this is what you get with crystalline. This is the isotropic phase. This is the nematic phase for the same LC. So I kind of went over the mechanism already. So what's happening is, what we think is happening, it's still not 100% clear, is that we basically diffuse the monomer in here. The monomer has to basically combine with another monomer on the surface. So you get some initial, basically, polymer oligomer hills, so to speak, formed on the surface that induces defects. And then these defect lines basically act as a guidance for the polymers to form. So if that's true, there should be some interesting things that are coming out of this. Number one, if it's really like that, then the elastic energy of the LC should really give you, influence the morphology and the diameter of the fibers, right? So number one. Number two, depending on where the defect lines go, we should get different shapes of fibers as well. So if you have, for instance, if you move from a nematic to a cholesteric phase, you should basically change that. That defect lines should get different shapes of fibers as well. So you should have both very nice control over the diameter and the shape of the fibers if those mechanisms hold. So this is one way to control basically the alignment. So one is homeotropically aligned LC, the other one is hybrid aligned LC, and you see very nicely that we do see a change in morphology of the fibers depending on what the alignment of the LC is. So that speaks towards the model that I described, and it's also telling us already, hey, we have some control over the shape here indeed. Now, with respect to the diameters, I'm looking at different LCs now. These are 3 different LCs with different elastic energies. And then, yes, indeed, we can control basically the diameter as a function of the LC. And in fact, it correlates with the elastic energy to some extent, at least. So this is a slightly different approach where we take the LC and we expand it over a mesh, metal mesh in this case. So the LC is sitting between the metal mesh here. It's probably organized perpendicular to the main surface here. And so what you see, the fibers are basically aligned too, but we are kind of lacking, we're making fibers, but we're lacking basically the order that we see in some of the other systems. Now we can then use functionalized polymers with this. So what we then can do is we can basically use this as arrays to immobilize biomolecules. So kind of the things that we have shown for flat surfaces with respect to immobilization of proteins and so on can be applied to these nanofibers, with the caveat, obviously, that the surface area is largely enhanced. So we get basically much more capacity, binding capacity for the proteins. So this might be interesting for some kind of highly sensitive applications. And you see here, essentially, an example where we then not only immobilize one functional group, but 2 by combining, essentially, by working with copolymers that combine 2 functional avenues that are orthogonal to each other, meaning they react only with one or the other target molecule. By doing so, we can basically then immobilize, co-immobilize 2 different molecules on the surface. In this case, these are just essentially 2 different fluorophores. So we get essentially the combination of colors here as well. So this is a very clear proof that we can co-immobilize those type of molecules. Now, I mentioned that maybe the nature of the LC plays a role here too. And so if that's the case, well, how about if we go to a cholesteric LC phase? So how about we go to a blue phase LC or like a smectic LC? And so we have looked at different types of LCs here. And in fact, you do see very nice changes in the morphology of the fibers as a function of the characteristics of the LC. And the nice thing, obviously, you know that there are a lot of different LCs out there, so we have quite a bit of flexibility in then essentially triggering different types of morphologies. So these are some that I really like. So this is cholesteric LC, but this time we're comparing a left-handed and right-handed cholesteric LC. So basically the dopant is has opposite chirality in both cases, and you can really see how that then impacts the swirling direction of the nanofibers here. So we can essentially then create counter or clockwise or clockwise helices with this process. Now, more recently, and this is not published, so I'm not going to go too deep into this, but more recently we started asking questions about, well, so if you look at this, right? So these are all achiral paracyclophanes. I mean, the paracyclophanes actually have planar chirality, but once we open them up, they are achiral, and then you have the chirality informed through the LC structure. But how about if we would actually introduce a chiral center into the paracyclophane? What would that do to the polymers? So that's kind of what we're doing here. So if we just do the nematic phase and basically a molecule that lacks central chirality, then we get straight fibers here. If we do the same thing with the chiral LC, we get this kind of swirling fibers. They're not really helices, I would say. They're kind of swirling because of the magnitude of the very large pitch that exists in those cholesteric LCs. Now, if we use a molecule like this that actually has a chiral center here and use a nematic phase, what we get is now this nanohelices that have very, very high pitches on the order of hundreds of nanometers. So that's really exciting. And so here, then as we change the EE of so basically we change the mixture of the two enantiomers in the paracyclophane that we create, we get basically different amount of pitches. So the pitch can be tailored towards, can be increased or decreased as a function of the ratio of the two enantiomers that we use. And also the twist angle changes as well here and steadily increases as the chiral paracyclophane becomes more and more pure. Okay, so there's more work to come here, and we have done a little bit more. We can actually induce chirality with those systems as well, which is interesting. But rather than going much deeper, I thought I'm going to take you on a little, towards the end of the talk, on a little kind of segue project that I'm very excited about, and just tell you a little bit about what can you actually do? Because I think many of you are thinking, okay, these are great polymers, but what can you do with those kind of polymers? And that's a project that one of my postdocs has been working on, and it starts very simple. So my question is, what is this? And so obviously we are on Zoom right now, but if we were in a real talk, you could guess. And I think most of you would say, oh yeah, it's a coffee stain, right? And so that looks like, right here on my desk, we have coffee stains and so on and so on. So why am I showing you this? Well, because I think it's actually physically very interesting. So if my surface was super defined, I had a very well-defined surface, I would deposit a very well-defined droplet, I would get something much more circular. Maybe I would get something that's entirely covering the surface, or I would get a nice ring, but it would be definitely symmetric. It wouldn't look like something like that. So the one thing we can do is we can make very well-defined surfaces. So let's see what happens if we do this following experiment. We're going to take a few droplets similar to the coffee stain. We take the surface that is very well-defined through our CVD process, but we add in this droplet, we add a little bit of a protein or a peptide as well. And so now you basically complicate the process because you also get protein crystallization here. And so you end up essentially with droplets that really look like this system here, okay? So it's much more heterogeneous. In fact, the patterns are relatively complex, and I find them just very intriguing as well. So where are those droplets coming from? So basically, obviously, we have a buffer system. We have a protein. So we have done a little bit of analysis. You can do SEM first order. You see the morphology. You see a little bit differences in the core region. Do you see probably what looks like a little bit more like salt crystals than you see in between region? You see some kind of a hybrid, and then probably outside you see maybe actually a little bit more of the protein it looks like. And so we can confirm that then with TOF sims as well. And we do really can track base some of those different regions to the different chemical composition that are either enriched of the salt that we use or enriched of the protein structure that we see. So there is a physical explanation for how those droplets those droplet patterns are forming. So now I would like to hypothesize that the position patterns formed on a material surface, on a well-defined material surface that is very homogeneous, such as the ones we make with CVD, during droplet drying are complex, characteristic, and reproducible. So with respect to complexity, there's something you can basically measure the entropy of an image, and then you can compare that to other entropies that are out there. So what we have done is we measured the entropy of the 30 most famous paintings that are ever painted and were selected by Forbes. So Forbes has a ranking of 30 most complex, most exciting paintings, most famous paintings. And then we measure the complexity of that, and we compare to the complexity of our drying droplet patterns over hundreds of droplets. And actually, they're not so different from each other. The painting's a little bit more complex, but our droplet patterns are quite complex as well. So we have complexity, and complexity for me means information richness, right? That's important. And then we also have the fact that they are reproducible, and then the topology is also influenced by the biomaterial, by the protein, and also by the surface. But the surface is something we keep constant here. So we get those kind of patterns here. This is another protein, looks quite different, but also very characteristic. We can make a lot of those droplets, right? So this is essentially now thinking about an automated pipetting system where we deposit multiple droplets, and you can see that they are actually quite reproducible, at least to the human eye. And then we can just go and say, let's do this massively parallel. So these are all, each of this is a wafer, glass wafer that has the size of a 96 well plate. And we just run them all through our pipetting system and create hundreds and thousands of those little droplets and create a lot of images, right? So this is great because those images, as I suggested, are rich of information. So images are information. So images of droplet stains are readily available. I think I convinced you on that. But what do you do with that, right? What do you do with those images? Well, what we have done is we started to run them through neural networks. And so those are neural networks that are pre-trained on images in the internet, humans, human faces, cats, dogs, I don't know. But they're quite well pre-trained. And then we use some of our droplets images to train them, about 1,000 to 1,500 for each run. And then we test them essentially with unknown images they have never seen. And we're asking questions about two things. We're asking questions about protein sequence and protein conformation, okay? So, and we decided to do this in one particular context. It could have been anything, but we got interested in amyloid beta because it's a key to understanding Alzheimer's disease, right? And so there's obviously, these peptides are involved in self-aggregation, automated plaque formation. That's very well established. What is not well established is the onset of that formation and how basically you will predict that at a very early stage. Because if you have those plaques, it's often already too late, right? So you want to really understand the impact early on. And we, again, as I said, we'll look at single amino acid mismatches and conformational mismatches. So basically, we take the primary sequence, we hit one amino acid and replace it with another one. That's actually relevant because a lot of the mutations that are pathologically active have one single amino acid switched out. And the other one we do is we're looking at misfolding of the protein. Okay, so this is the first thing, single amino acid mismatch. This is the peptide. You have the sequence here, and then we go in and say, okay, we take the second amino acid and we replace it with either a V or a T. So we're basically looking at those different structures, and this is the images we collect. And so you'd say, okay, maybe they are quite different in some cases, but they're very similar in others, right? So, and then of course you also have to consider variability within a single droplet system. And so now we're basically running that through our neuronal network. First of all, let me show you the physical properties. So molecular weights are essentially identical. Isoelectric points are very similar, except maybe the lysine 22. The hydrophobicity is very similar. So it's very hard to make a lot of sense out of this just from a physical analysis. So when we run it through our neural network, we basically can predict so first of all, we say that they cluster very nicely in our Disney plot, so they're very different from each other. I think that's what this suggests here. And then when we basically run the different test images, so these are images that the network never saw against training images, we get to predict the composition of the peptide with 98% accuracy. So we can, with 98% accuracy, predict which mutation happened in this particular protein. And the ones where we basically miss it, or the network actually misses it, so we always give the fault to the network. In those cases where the neural network misses it, we can go back and then understand why it missed it. So, okay, so now, sorry, I took multiple ones. So this is basically how different networks learn. So this is also interesting for us because it helps us. So this is the droplet. It helps us. This is the same droplet in all cases. And so it helps us to understand which parts is it picking up on. Is it picking up more on the core, which is more of the salt component? Is this more in the hybrid area, or is it on the rim, which is more protein or peptide rich? Okay, so it works well with sequences. How about conformational changes? That's even harder, right? Because those are a little bit more transient potentially. Now, the nice thing is the field is very well versed in inducing those conformational mismatches. And so what you do is you take the peptide and you add a little bit of a solvent. It's called hexafluoroisopropanol in there. That creates a hydrophobic environment. And then basically by the amount of this HFIP you add, you basically then can disturb the structure and the alpha helix and beta sheet content systematically as a function of the amount of solvent that you use. And so by doing this, we have access to different variants of this, or different conformations of this particular peptide. So the sequence is always the same, but the conformational state changes as a function of the HFIP. Now, that's okay, but the question is this maintained when we then basically put it back into a normal buffer system? And so we basically treated this for 15 minutes, took it back in our buffer system, and then measured the CD data that you see and the alpha helix beta sheet content in that particular buffer. And so that works actually quite well. And you can see that we can change this systematically here, and then you see the droplets that we created. And so the number here is basically always the helical content of the peptide. So the higher the helical content, the more structured, the more normally structured the peptide is. So as we go to helical contents below 10, for instance, it becomes quite disordered. Okay. So now the question is, how well can we then predict the helical content of that peptide? And so we created 8 different variants again. We compared them with each other. And while there are a little bit more kind of mismatches here potentially, overall, they still cluster very nicely. And the accuracy in this is again, very, very high. So this is really exciting because now you can actually really not only predict a structural difference, but you can also predict misfolding within those peptides. Okay, so this is where we are right now. We have confirmed, we are confirming some of this a little bit more and so writing a manuscript on this. And then the next thing is to look at maybe even protein-protein interactions, protein-DNA interactions, bringing it into other areas of biomolecular analysis potentially. And so let me just conclude here by the lessons we've learned so far with this last project. So first of all, this droplet stains, even though they're experimentally very simple to access, they're actually quite complex. And so they're not only literally complex as you measure the complexity and the entropy of those images, but they're also complex in the way the process that involves to form them. But they do depend on the protein that you use, the peptide, and they depend on the surface as well. So we have a similar set of data where we look at different surfaces as well, and you can actually use this as a way to predict surface composition as well. The other thing is existing, and we do. My lab is not a deep learning or machine learning lab. We use existing machine learning algorithms out there. We can repurpose them to accurately predict very intricate properties of those peptides. And then we think that we can actually expand this to interesting protein-DNA interactions, protein-protein interactions, certainly surface analysis as well. You might be able to predict protein aggregation tendencies in those systems as well. So we're very excited about this. And again, to put this all together, it's really enabled by the ability for my lab to control surfaces very precisely through the CVD. Because one thing I can tell you, if the surfaces are different, the neural networks will pick that up as well. And so that will be an interference for sure. So with that, I'd like to thank Kenneth Chang, who is basically the student that started a lot of the CVD work related to nanofibers, Ramia, surface modification. Asam is the student that did all the neural network and drying droplet work. Divya is a postdoc that has created the last part of my talk related to this helical nanofibers. And then John and Xiao Yang are newer PhD students in my group that are working on CVD. So is Arit, who is starting to contribute as well. So with that, I'd like to thank you for your attention, and I'd be happy to answer any questions you have. Thank you.
Thank you, Dr. Lohan, for the talk. So we have some questions in the chat. So the first one's from Sophia. It says, have you considered looking at the way these different surface topologies can direct cellular expression and possibly tissue engineering?
Yeah, I think that's a great question. And also chirality, right? I mean, what is the role of chirality as it approaches a cell membrane? Especially then if you dissect between bacteria and mammalian cells. I think that's a really exciting topic. We haven't done a lot of that. I think you will have multiple effects. I mean, certainly you have a surface area effect, you have a topological effect, and then maybe you have secondary effects from chirality. Something I really want to do. We haven't done it yet, but we should.
So the next question's for me. What's the time scale of the CVD polymerization, and can you like actively change the orientation of the liquid crystals with like an electric field while it's polymerizing?
Yeah, great question again. You guys are doing. I don't have to write any grants anymore. You guys were, I think, giving me all the good ideas. So the time scale is. So I mean for the thin film, so anywhere between a few minutes and 10 minutes, I would say, to get, depending on how thick you want the polymer coating to be. So we deposit at the level of a couple of angstroms per second at steady state. So it goes relatively fast. Of course, it takes time for the system to be evacuated. We work in vacuum and so on. So if you take a whole run, it's more like 30 minutes. So the second idea is something we're doing. So can we change essentially the LC during deposition, or can we change can we apply electrical fields to the fibers in the conjunction of while still in context of the LC, and can we then bend the fibers potentially, right? So those are related, a little bit different, but related things. So we're doing that right now. There's a paper that's going to be coming out probably within the next half a year or so. But it works in principle, so it's a good idea, yeah.
Okay, I don't see any other questions. I'll just ask another one from me. What's the overall structure of the neural network? So do you start with the image pixels and then finally map it to softmax probabilities of like the sequence or different classes for the secondary structure, tertiary structure?
So let me see. So these are image algorithms, right? So they don't really know what is what. They basically we take this pre-trained set, right? So they're basically coming out of, this is GoogleNet or something like this. This is what you see in the internet as well. Then we train them with about 1,500 to 2,000 training sets. So we basically, when I showed you this 80 or so plates, all of those are the particles that we use. Then a subset of this is used to train. So the neural networks looks at essentially image and says, well, those are similar, those are similar, those are similar. And then we offer test droplet images. And then it says, oh, that image is similar to that. That image is similar to that. And so that's then how it makes the connection. So I don't think the network knows what the origin of the droplet ultimately is. It just looks blindly at images and tells us similarities.
Okay, so it tries to like cluster similar images, kind of.
Yeah, exactly.
Are those all the questions, Azam?
Yeah, go ahead.
Okay, great. So thank you so much for a wonderful talk. I really apologize for the late start. There was a bit of miscommunication in regards to the person hosting for today. So I appreciate all of your patience. So next we're going to transition and Leah, I'm not sure if that's how you say your name. I'm sorry if it's not, but she will be presenting Professor Stashi.
Okay, hello everyone. My name is Liat Kogomas, and I am a PhD candidate in Professor Aaron Stashi's lab at Cornell University. I'm very excited to be introducing Aaron today. Professor Stashi received her bachelor's in chemistry at University of Wisconsin, Green Bay. She then went to pursue her master's at Colorado State University with Professor Eric Ferreira. She received her PhD in chemistry with Professor Tomislav Rovis and Professor Abigail Doyle at Colorado State University. Her career as a polymer chemist really kicked off as the Cornell Presidential Postdoctoral Fellow with Professor Brett Fors here at Cornell. Now, an assistant professor of chemistry at Cornell University, Professor Stashi has received various awards and grants, including the President's Council of Cornell Women Affinito Stewart Grant. Professor Stashi's group researches polymer chemistry with a sustainability focus, and today we'll be talking about photon-driven strategies for depolymerization. Personally, I can say that Aaron is a brilliant scientist and a fantastic mentor as well. With that, I ask that you please turn your attention to Professor Stashi as she begins her talk. Thank you.
Thank you, Liat. That's a very kind introduction. I hope everyone can see my screen and hear me okay. All right. I didn't see anything to the negative, so that's good. All right, so today I'm going to be talking about some brand new research coming out of our group. As Liat mentioned, we just started the group up about a year ago, so everything here is brand new and unpublished. So hot off the, hopefully soon to be published, but be hot off the presses. But first, I want to thank all the organizers, especially the graduate students, for putting this seminar series together. I know the students in my lab have enjoyed watching this all summer and seeing all the amazing research going on across the country. And I think this is just a great opportunity for people to showcase some of the science going on in their labs. So great job, and thank you so much for putting this together. So as Liat introduced me, she gave me a little bit of background on me. I like to talk about this at the beginning of a talk, particularly when there are a lot of graduate students and postdocs in the audience who may be thinking about a career in academics, and sometimes my sense is that people feel there's only one way to get here. It's bachelor's degree, PhD, postdoc, and then off to be an assistant professor. I absolutely did not take that route, made it a little bit more complicated for myself. Like Liat mentioned, I got my master's degree instead of finishing my PhD at Colorado State, because I wasn't quite sure if I actually wanted to get a PhD at the time. I wasn't sure what I wanted to do with my career, my life. So I worked in industry for a while, and that was really informative and gave me a lot of perspective, particularly working at HRL Labs. If you haven't heard about this company, They're in Malibu, California, so a great location, and they do some really interesting research. They're owned by General Motors and Boeing. They do a lot of automotive and aerospace applications. But working with polymers and materials for the first time, I loved the research, and it really pushed me to go back and finish my PhD. And then from there, I finished up being a postdoc, and then loved Cornell so much that I got to stay here, which was great. But I just want to say that there's no right way to get to where you're going. Just work hard and be passionate and excited about what you want to do, and I think everything will work out well. So, but that being said, so when I was working in my PhD and my postdoc, a question that I would frequently ask is how can we use light to access new reactivity? There's just been a boon of photoredox catalysis over the last, 15 years now. I need to change my spiel a little bit. And it's just given us so many opportunities to access new reactivity, new bond forming reactions, new bond breaking reactions. And in particular, in my PhD, I used photoredox catalysis to build complex small molecules, either things that would map on really well to natural products, or developing new methods for hard to activate carbon-oxygen bonds or other types of bonds from feedstock chemicals that could be then converted to more complex materials. And similarly, when I came here to Cornell to work with Brett, we thought about using photoredox catalysis to make advances in polymer and material science, taking carbon-hydrogen bonds, which are ubiquitous structures, to polymers, and grafting off of them in a hat raft strategy, which Brett talked about a few weeks ago, so I'm not going to talk about any of that today. But the question going into my independent career was really the same thing. And what I think about and how I base all the projects that we carry on in my lab is really thinking about photocatalysis and how we can use light to develop new reactivity patterns and think about making huge advances in material science. But the other half of that is we're very much focused on sustainability. We certainly have a plastics crisis looming, and we're thinking about how we can make biorenewable plastics, biodegradable plastics, but how we can also tackle some of the existing materials that we've spent 100 years perfecting the synthesis of, but now we have to find a way to get rid of them as well. And so they always say problem selection is a really big part of research. So we went after a very big problem, that is polymer recycling. Many of you probably already know about this. This is a study that was done a few years ago, where they tracked all the plastics that have ever been produced. 8,300 million metric tons of plastic have been produced. 70% of this has actually ended up in waste streams, in landfills, in the oceans, in animals, in us. Some of it's burned, but very little of it is actually recycled. And we look at the most frequently used commodity polymers like polyethylene and polypropylene. We make these on over 100 million metric tons per year, and this is still increasing, and yet we don't really have a way to recycle these. And so in thinking about recycling, what are the strategies that we do have, or how do we think about taking polymers and turning them into, at their end of life, turning them into something useful? And so for the life cycle of a polymer, most of our polymers, commodity polymers, come from petroleum feedstocks or oil, can be refined into the monomers and then polymerized and then either molded or extruded or blended with some other material to make the products that we use. Hopefully we use these products more than once, but in a lot of cases it's single waste plastics, and then they end up in the trash. The methods that we do have for recycling are things like mechanical recycling, and this is separating out the plastics, chopping it up into smaller pieces, and then remolding it and extruding it into a new product. Now, this is a really good strategy for things like polyethylene and polypropylene, but there are a lot of challenges with this as well. And for one, separating different types of plastics. Once they all get mixed together, it can be harder to separate them based on their chemical structure and density. And the other thing is mechanical recycling has a finite life cycle. You can only mechanically recycle a polymer so many times before you start to lose some of the physical properties. You start clipping off the chains, and as you get shorter and shorter chains, you start to lose some of those really important durability properties. And so the ideal way to recycle a polymer is chemical recycling. And so this would be taking a polymer and reducing it back down into its monomer starting point. That way you can make it into a brand new polymer with the perfect properties that you want, or you can repurpose that monomer however you like. And not to give away the punchline too much, but we're thinking about doing this with lights. This is a booming area of research. I think a lot of groups are interested in this. In this department, Jeff and Brett have recently published some chemical recycling strategies. Eugene Chen, Jeff Moore, and McNeil, among a number of different people looking at this. But this is where we also want to make a difference in polymers and materials. The challenges here are identifying depolymerization strategies that are going to be compatible with waste streams, because again, when we recycle things, they don't always come in nice, clean plastic like we make in the lab. They tend to be mixed with plasticizers, composites, other polymers, and waste and detritus. And also developing a generalizable strategy. So mechanical recycling, that's something that you could use across a number of different polymer types. But for chemical recycling, polymers are very, very different. And if we look at just the diversity of the commodity polymers that we use, looking at our resin identification codes, things from polyesters to polyolefins. Polyvinyl chloride has unique challenges, generates HCl when you try and depolymerize it. Polystyrene, and then the catch-all, which is everything else: polyesters, polyurethanes, polycarbonates. How can we develop a generalizable strategy to try and depolymerize all of these in a chemical recycling strategy? And we ask the question: how can we use light to depolymerize such a diverse set of polymers? So we're going to put a pin in the light for just a second. Heat is a general depolymerization stimulus. And so we all know that polymers are susceptible to thermal degradation. When we think about thermal degradation, we think about kinetics, we think about thermodynamics. We look at some of our commodity polymers here: polyethylene, polystyrene, PMMA, and polyalpha methylstyrene, which we kind of use as a reference polymer in our lab. So kinetically, these polymers are all trapped when we make them. They don't have any reactive sites on the polymer backbone, and they all have specific degradation temperatures. So if you heat the polymer above this degradation temperature, you're going to start to break bonds in the backbone of the polymer and generate reactive species like radicals or ions that can then go on to do either cross-linking or depolymerize to give you back monomer. And so polyethylene has a much higher degradation temperature, pretty unsubstituted, and as you add substitution, the degradation temperature starts to go down. Now, the other thing to think about in polymers is the thermodynamics, and this is the sealing temperature. So all polymers are unstable at a certain elevated temperature. Most polymers are unstable at an elevated temperature. And so the sealing temperature is the thermodynamic principle for actually turning monomer into polymer. Polymerizations are driven by enthalpy, but they're are going against entropy by stringing all of these small molecules together to make a long chain polymer. And so at a certain temperature, that entropic cost will outcompete the enthalpic benefit, and you can actually push the polymerization towards depolymerization. So you can think about this: if you're below the sealing temperature, you can make polymer, and if you're above the sealing temperature, you can depolymerize it to monomer. Now, these are absolute sealing temperatures. There are actually an infinite number of sealing temperatures for every polymer, depends on concentration. But at these temperatures, you cannot push monomer towards polymer. And so this is how we're starting to think about depolymerization of polymers. Can we use heat as a stimulus to do this? So it turns out if your degradation temperature is far below your sealing temperature, you're not really going to push this back to monomer, because you're just going to keep breaking bonds in the backbone, and you're just going to get a lot of side reactions. But as you get closer to merging these two numbers, as your sealing temperature gets closer to your degradation temperature, you start to get monomer back. So with polystyrene, you can get almost 50%. And as you move to these other polymers, you can get almost quantitative return of your monomer if you heat these high enough. But I said we wanted to use light. And so the concept that we were really intrigued about was photothermal conversion. And so this is using light and nanoparticles to generate thermal gradients. And so you can imagine if you have these nanoparticles in solution or in solid phase, and you irradiate them with visible or near IR light, these will generate temporal heat gradients. And so you'll get really, really hot temperatures right around the surface of the particle, but the bulk solution itself will stay around room temperature or slightly higher. And so if these temperature gradients interact with a polymer, you can imagine that you can start breaking bonds in the backbone of the polymer. And so the way this works is through, well, the way this works for many of these nanoparticles is through surface plasmon resonance of metallic nanoparticles. And so these are things like gold and silver and copper oxide. And what happens is you get a collective oscillation of the conduction band electrons when they get photoexcited. These are in resonance with the incident wavelength of light, and this creates a very large cross-sectional absorbance, which then when it relaxes back down, gives off heat. And so when we're talking about giving off heat, in the case of gold, this can actually reach temperatures of about 1200 K. This is based on the fact that gold melts at 1200 K and it starts to actually coalesce and lose its form. So these things get really, really hot if you hit them with enough photons. This isn't just exclusive to plasmonic materials. You can also do this with other photo dyes and things, anything that will absorb light. If it relaxes back down in a non-radiative fashion, it can give off heat as a byproduct. And so we compare the ideas of bulk heating. This is taking something in solution or in solid phase, giving it enough energy that everything, including its surroundings, gets up to temperature. So if you're running a reaction at 100 degrees C, it's not just your reagents that are at 100. You need the solvent to be at 100. You need the oil bath to be at 100. You need everything to get that hot. You get a very uniform temperature across the entire form. Photothermal heating is very different. In this case, you're only getting heating at the surface of these nanoparticles, and it decays pretty quickly as you move away in nanometers. And so these are basically little nano heaters. And so you have all the temporal and spatial control of visible light, but your active stimulus here is actually heat, which is just a super cool concept. Another way to think about this, and if you take nothing else away from this talk, you should go check out this paper because it's really funny. The opening line just cracked me up. But it's a good macro scale explanation of plasmonics and how these work. Apparently there's a whole YouTube phenomenon. You can put grapes in the microwave and put them together and they'll start to form a plasma in the middle. And so this just kind of gives you a general idea of how microwaves, which are very long wavelengths of light or electromagnetic radiation, come in on these macro scale particles, which are basically representing the nanoparticles. And you get this collective oscillation. You can get this plasma to form in between these particles. And in a lot of plasmonic applications, you're getting the highest temperatures at the union of two particles together, although you don't need to have two particles together. Some of the other applications of the photothermal effect have been used in photoablation of tumors, so heating the tumors to try and denature the proteins, but also water purification. And so gold and silver and copper oxide have been used to purify water. And so the idea is that you radiate the solution of the nanoparticles in water. It'll superheat the surrounding area around the particle and denature proteins and get rid of viruses and bacteria and things like that. But it doesn't boil off all the water and have it end up in the air. So it's a really cool way to purify water in like third world countries. There is some photothermal depolymerization precedent. And so Ben Lear at Penn State published this paper a number of years ago now, where they were able to degrade polypropylene carbonate using gold nanoparticles and laser intensity pulses of light to form volatile components like and carbon dioxide. And so in this case, they were radiating a very small film of polypropylene carbonate with these nanoparticles embedded, and they saw they could get about 80% mass loss with increasing gold fraction, which is a really cool application and demonstration that you can use photothermal heating to do depolymerization or at least polymer degradation. Now, the limitation of this approach is one, it's using gold, which is very, very expensive and probably not the best strategy for degrading commodity polymers. But two, like I said before, gold is not the most stable. It exhibits a very extreme LSPR, but in doing so, it generates really hot temperatures, which can actually melt the surface, and the particles start to aggregate. And once they start aggregating into these larger forms, they lose that surface plasmon resonance, and then they're no longer as effective. And so with this in mind, we thought that we could use photothermal materials to try and degrade commodity polymers, or do depolymerization as a function of this photothermal heating concept. And so the idea is to take a polymer in the presence of these nanoparticles, irradiate it with visible light, and recollect the monomers, which can then be used for polymerization again. So the questions that we asked are, can we do this? Not using laser intensity light, can we use standard LED setups that we're used to using in photoredox catalysis all the time? Can we use that less intense light to still get photothermal heating and affect depolymerization? What types of plasmonic materials are actually going to be compatible with the kind of polymers we're looking at? And can we develop protocols for predictable temperature gradients? Can we say, okay, we want to get to this temperature, this degradation temperature. Can we get the right material? Can we get the right wavelength? And can we get the right intensity? And then can we tune that so we might be able to do selective depolymerization of polymers based on their degradation or sealing temperatures? So this is what we set out to do, which is a very, very ambitious idea. And to start, we wanted to pick a model system for depolymerization, something that was well behaved with depolymerization, so we could really study the photothermal heating effects using our particles and our lighting setup. And so we picked cyclic polyethylaldehyde, which has been extensively studied by a number of different research groups. Anne McNeil, who gave a talk last week, although I don't think she talked about this, Jeff Moore, and a number of other groups. It's a self-immolative polymer, and so when you heat it up above its degradation temperature, it tends to depolymerize back into monomer. Depending on the substitution pattern, which is what we really liked, we can change the degradation temperature. And so if we put more electron withdrawing groups on here, we can raise the degradation temperature, which now gives us a gradient of temperatures to look at. And this also changes the sealing temperature. To make these polymers, we've been developing a few methods on our own, but also using some of the literature precedents. Usually just take a Lewis acid and you can make the cyclic polyethylaldehyde. Now to depolymerize, a number of different stimuli have been used. It's not stable to acid, so throwing in some strong Lewis acid or Bronsted acid will depolymerize it pretty quickly. You can heat it up above its degradation temperature and it'll depolymerize. You can sonicate it and break the polymer up that way. And most recently, Jeff Moore showed that you could use visible light and generate or do single electron transfer to oxidize the polymer and push it towards depolymerization. And so they showed that they could take a film of the cyclic polyethylaldehyde with this acridinium photocatalyst, which is very oxidizing, and in the presence of plasticizer, they could affect a single electron oxidation, and mesolytic cleavage will ultimately cationically unzip the polymer to give off monomer and destroy the polymer film. And so this is what the films look like. And so in just 24 minutes, they pretty much completely destroy them. This very pretty yellow neon color of the acridinium, and then it decomposes over time. The important thing to note here is that cyclic polyethylaldehyde is not stable to UV irradiation. So if you leave it in sunlight, I don't want to give too much of a spoil away, but it's not going to be stable. And in this case, when they're running these depolymerizations, they're using 370 nanometer light, like LEDs. But if they put it in sunlight, they can affect the same depolymerization in about the same time scale, actually. So it's a very efficient sunlight-mediated depolymerization. The way the depolymerization of polyethylaldehyde works, I kind of just showed you, but cyclic polyethylaldehyde, it's thought that you can open up the cycle, extrude a monomer, and then it can actually close back up. So you might need to open and close it several times to get it to depolymerize. And as you might expect, one way to track this, one is monomer formation, but the other is watching the GPC traces. The molecular weight of the polymer should decrease as you convert it to monomer. And so without any monomer, you should have a very large polymer, and then it will decrease over to nothing by the end when you have 100% monomer. And so with all this in mind, we have our system, we have our polymer that we want to use, and now we want to ask these questions. So what material, what photothermal agent are we going to use? I've already said that gold is maybe not the most practical for doing large-scale recycling of commodity polymers. And so we turn to something that's already used in polymer chemistry pretty frequently, and that's carbon black. And so this is a very inexpensive photothermal agent. You know, we spent 20 cents a gram from Alpha, but it's much cheaper than that. It's a product of petroleum refinery, and it's already used in polymer chemistry as a filler, as a pigment, as a stabilizer, a UV stabilizer. It's a broadband absorber, so that means we should be able to hit it with multiple wavelengths of light and perhaps generate these temporal heat gradients to depolymerize cyclic polyethylaldehyde. And so all this work was carried out by Liat, who just introduced me before. So she's a rising fourth-year graduate student in the lab, and she's responsible for everything that I'm going to be talking about today. And so what she did is she first synthesized some cyclic polyethylaldehyde, and then in the presence of carbon black in solution, you can suspend it through sonication, add in the polymer, expose it to red light, and after just 4 hours, we see 100% conversion to monomer. So she took this 24K polymer and converted it cleanly into monomer. We did some control experiments, you might expect, and there's no conversion in the dark and there's no conversion without carbon black. So even though these red lights get kind of hot, they're not getting hot enough to do solution depolymerization of the polyethylaldehyde in the absence of carbon black. But we did want to compare the kinetics of depolymerization as a function of photothermal heating versus bulk heating. And so we just took the polymer and heated up in solution, just in the absence of carbon black. And what we found is that once you get above 90 degrees C, that's when you actually start seeing depolymerization, solution phase depolymerization. And once you get to 100 degrees C, after 4 hours, it's completely depolymerized, although not cleanly. There's definitely some degradation of the monomer in solution. Now, in comparison, we've rudimentarily tried to estimate the temperatures, the bulk temperatures that we're reaching through the photothermal effect. And we do see an increase in temperature above room temperature to about 67 degrees C when there's carbon black in solution. But that's still significantly lower than this bulk solution temperature that we need to get effective depolymerization, which means that the surface of carbon black must be much hotter to actually affect the depolymerization in this 4-hour time scale. So we started with solution phase because that's easiest, just dissolve the polymer, add some carbon black and call it good. But in practicality, we don't really want to dissolve all the polymer and then irradiate it with light in solution, especially not in DCE, which is a chlorinated solvent. And so now we're looking into solid phase depolymerization. And so Liat learned how to cast some different films with and without carbon black. And so in this film right here, you can see this is just the polymer on its own. It's very clear and transparent, although a little brittle. And then this is what it looks like with just 1 weight % carbon black. We've gone lower to like 0.5%, and it's still sort of midnight black, but very, very well dispersed and well suspended in the polymer film. And so the question is, all right, is this still going to work once we move to solid phase? It worked really well in solution, but you have a lot of chain mobility. So we weren't entirely sure if this would work in solid phase. And then what is the carbon black going to do to the physical properties of the film? It's going to destabilize it, or is it actually going to improve it? And so she took this film, a cyclic polyaldehyde with a few different weight percents and different conditions of carbon black, and exposed it to red light irradiation. And so usually what she does is she cuts off a little piece of film and then sticks it in a vial in front of the light. And with 10 weight % carbon black after 3.5 hours, it's pretty much depolymerized back to monomer very cleanly. We just see polymer and monomer in the NMR, about 75%. Interestingly, as she decreases the amount of carbon black to just 1 weight %, it's actually even more efficient. And this kind of makes sense. Carbon black absorbs most of the light. It's a black body. It's almost a perfect black body, so it's absorbing all the light. If there's too much in there, it might actually be blocking transmission into the film. And so with 1 weight %, we actually see more efficient depolymerization. And we can push this to about 100% if we let it go for a little bit longer. Sometimes what we see happening is that the carbon black starts to aggregate as the film is depolymerizing, and then that just takes a little bit longer to ultimately depolymerize. Now, most commodity polymers are not pure polymer. They usually have other composites or plasticizer in them. And so Liat added some diphenyl phthalates, 10 weight %, and we still see pretty efficient depolymerization, albeit a little bit slower, but it still depolymerizes back to monomer very cleanly. And the control, if there is no carbon black, we don't see any depolymerization, sorry. Even though it's getting pretty hot inside of those vials, the films just look completely perfect, just like they did before we irradiated it with red light, suggesting that the photothermal effect is responsible for depolymerization. And so these are what they look like in practice. So this film on the left here is the film without carbon black in it. You can see it's pretty much unchanged. It still has its structure. It hasn't melted. It's not yellow. And then when there's carbon black, these are cast from 2 different solvents. You can see that as it depolymerizes, the monomer actually is melting and sublimating onto the surface of the glass. So we could actually collect this and repolymerize it, most likely if we wanted to, just something we're still working on. But you see the polymer is, or the film is pretty much completely disintegrated. Now, the advantage of using photothermal heating versus bulk heating is that you should be able to get spatial control. You should have all the spatial control of visible light and the temporal control of visible light. And so we question whether or not we could take a film of the polymer and the nanoparticles and expose it, just part of it, to light. Would we be able to see the spatial resolution and only see depolymerization where we're shining the light? And so we made a very crude photomask just to start. This is just electrical tape over a scintillation vial, and this semicircle represents the film, 1 weight % carbon black. And then after irradiation, you can see that the film has depolymerized. You can see that this is all spotty right here. This is still covered up. And when we uncover it, we can see that we get pretty good spatial resolution. Over the film, it's depolymerized where we're shining light, and it's intact where we are not shining light. Now, this is not the best photo mask, and this isn't collimated light or anything. So this is actually really impressive spatial resolution. And we're working on some other more complex techniques to show that we can do this, which we think we definitely can. So we've done solid phase depolymerization. We don't have to have this in solution. We can just make a film and then irradiate with light, and it will depolymerize. But what does the carbon black do to the physical properties of the film? You can imagine that if you wanted to use this type of strategy for polymer recycling, it would be ideal if you could incorporate the nanoparticle into the polymer as you're formulating your product, because then you just have the trigger built in. But that's only going to work if it doesn't erode the properties of the film, and it won't depolymerize on its own when you're trying to use the product. And so we're starting to look at the physical properties, tensile strength and things like that. But mostly we want to see, is the polymer film going to be stable if it has carbon black in it? And so we're using the Ithaca sunlight. We want to see what would happen over long-term exposure. And so we took a film of cyclic polyaldehyde without any carbon black, and then we have a film that has 1 weight % carbon black in it, and just set it on the windowsill for an extended period of time. We get very good sunlight in the afternoons on that windowsill. It gets very, very hot actually. And after 12 days, you can see that the polymer is starting to degrade if it doesn't have any carbon black in it. It's starting to yellow, it's starting to wrinkle up, and starting to shrink. And then after 84 days, almost 3 months, it looks, really disgusting actually. It doesn't really look like a film anymore. It's turned yellow and it's kind of gooey. It's just really, it's not a polymer. After 84 days with carbon black, it seems like it's slightly more brittle. We still need to actually get a number on those physical properties, but the film itself is still intact. What's actually still in the film, so this is a polymer that she used to make the film, and with the absence of carbon black, all we see are oligomers. There's nothing in the GPC trace, and in the NMR, it's mostly degraded monomer. And so it's not even phthalate anymore. We can't recover it. So this polymer is not stable to sunlight. It's really not stable to ambient light for a long period of time. Carbon black, however, we've seen a small amount of degradation, but this actually looks pretty good. By the NMR, we see less than 1% monomer, and we don't see any UV degradation products. So the carbon black is actually protecting the film, protecting the polymer from degradation. It's actually stabilizing it for really long periods of time. This is actually a very long lifetime for this polymer, just out in ambient conditions. Normally, it's not going to be that stable. But the question is, can we still trigger depolymerization when we want to? And so we took this film that had been sitting in the windowsill for almost 3 months, hit it with red light, and we get clean conversion to monomer with a little bit of oligomer left over. You can see down here, this is the experiment that Liat ran, where the monomer has collected on the glass as it depolymerized, and there's just a little bit of aggregated carbon black and polymer left in the bottom that wasn't really getting hit with light anymore. So it's a really cool experiment to show that the carbon black is actually stabilizing and giving this polymer a much longer lifetime, suggesting that this is a material we could embed into our polymers, use the product, and then just trigger depolymerization when we want to by shining more intense light on it, because sunlight itself is not enough to induce enough photothermal heating to get it to actually depolymerize. So we can depolymerize solid phase films, and the carbon black actually improves the physical properties of the film, which is really exciting. And this is something we're going to look at as we explore other photothermal materials as well. And so lastly, the thing we're starting to work on now is doing selective depolymerization. And so based on the degradation temperatures and the sealing temperatures of these polymers, we should be able to hit a specific temperature gradient and get selective depolymerization. And so for example, cyclic aldehyde decomposes about 110 degrees C, whereas polystyrene and alpha-methylstyrene have much higher degradation temperatures. And so if we mix all of these together in solution with the carbon black, we should only see depolymerization of the phthalate. And so this is the phthalate polymer that we started with. This is the polystyrene. This is the polyalpha-methylstyrene, very broad sample. And then when we subject it to the reaction conditions in solution, we only see depolymerization of the cyclic aldehyde. You get 100% conversion to monomer, and you can see that the polystyrene and the alpha-methylstyrene are still intact, and they have not been depolymerized, and we don't see any monomer in solution. And so now we're moving on to solid phase. Can we make films of these polymers, which of course, if they're not copolymers, they're not going to phase mix. We're trying to develop ways to get these all into solid phase so we can test the depolymerization as a mixed stream. And so the ongoing research directions for this project, we're working on more high resolution spatial controls. We're using collimated light and photomask. We have some really exciting results, I think, coming out of that. Looking at solid phase mixed stream depolymerization, looking more at the physical properties of what carbon black does to the films, and seeing if we can get some specific numbers on those, and then showing that we can depolymerize and repolymerize the polymer. So if we take the polymer, depolymerize it, we can collect the monomer and then resubject it to the polymerization conditions. And we should also be able to do this with carbon black, which is something we're also going to look at. And so with that, I need to thank the people who have actually done the work. This project is entirely started by Liat, a fourth year in the lab. It's her right here, engaging in some shenanigans with the rest of the group during the 1 month when COVID was pretty down here in Ithaca. But I want to thank the entire group. It's been amazing in getting everything started, and we're doing some really cool stuff. Hope we can talk about more of that in the future. I want to thank my incredible mentors here at Cornell. Brett and Jeff and Tristan have been really, really supportive and helpful. Cornell for funding, and the President's Cornell Council for Women for giving us funding for this project. And please follow us on Instagram and Twitter. Sometimes we post stuff. So thank you for your attention, and I'd be happy to take any questions.
That's a fantastic talk. Thank you, Erin. So I will start by asking some questions, and if we get any in the chat, I'll read those out as well. So I was wondering if you've thought of different ways to increase the efficiency of heat transfer, especially in those solid films for polymer.
Yeah, so as you know, the reason we started with solution phase depolymerization is that it was easiest to get the polymer dissolved and get the particles suspended in solution. And what we found is that the polymer actually, the carbon black suspends really well with the polymer, but it actually starts to crash out of solution as we depolymerize it back to monomer. And so that's why we moved to the solid phase, because we thought we'd have better heat transfer. Because in solution, you're going to have a lot of heat loss just to the solvent, and it's not going to be used to actually break the polymer backbone. And so now we're looking at ways of both qualifying and quantifying the degree of interaction between the carbon black and the polymer itself, looking at different ways of functionalizing the carbon black, and trying to get more consistent size. Because right now we're just using bulk carbon black out of the bottle. Imagine if we can get a more consistent size, it might suspend better and give even more efficient heat transfer.
We have a question from Luis.
Hey, Aaron. I don't know if I'm just like not grasping this concept, but if you're achieving such high temperatures, what about the localization of those temperatures makes it so that you have fewer side products? Like, why aren't you having those same sort of oligomerizations or side reactions that you would have if you just raised bulk temperature by localizing it? Like, I'm sorry if that's like totally basic and that's just going over my head, but
yeah, no, I think that's a really good question, and I'm trying to find a really good way to describe that phenomenon. It's something that we've seen in our work. It's something that other people have used in or seen in their photothermal conversion. Higher temperatures leading to much enhanced reaction rates, but seeing really clean products at the end. And I think, with bulk heating, you're keeping these products at an elevated temperature the entire time, versus when you have localized heating, it's only getting heat for a very short amount of time. I guess I kind of think of this analogously to photoredox catalysis. When you're doing electron transfer, you need to have that excited state photocatalyst get very close proximity to the substrate you want to activate. And if it was constantly hitting that, you'd probably get a lot of, if you were electrifying the entire solution, you would probably get a lot of degradation and see a lot of byproducts. But when you only have these short interactions, I think that's why you start to get much cleaner product distribution. It's because it's not at that elevated temperature for extended periods of time. So it really depends on, what the degradation mechanism is, what sort of energy does it need, what is the activation barrier. I think thinking about those things.
So the next question in the chat is, what is the importance of sealing temperature and depolymerization temperature when trying to degrade polymers?
Yeah, so I like using alpha-methylstyrene as an example. Well, actually, no. The cyclic polyaldehyde we're using is an even better example. So that has a sealing temperature of -36. And so when you're trying to polymerize it, you cannot polymerize it above -36 unless somehow you were trapping it after every single monomer addition. But if you have active chain ends, it won't actually propagate. It'll just go back to monomer. And so then when we want to isolate it, we throw in some base to quench the acid, and then the polymer is kinetically trapped. And it is stable, even though it's well above its sealing temperature up until, I think, the degradation temperature, which is 110 degrees C. So then once you reach 110 degrees C, that's when you actually start breaking the bonds in the backbone. And then now you have reactive sites, and so that can push it all the way back to monomer. And so when you have the sealing temperature below your degradation temperature, you can actually push that, thermodynamically, you can push that equilibrium back to monomer. Now you also have to think about the kinetics of this, which is something we're also looking at. Can we accelerate the depolymerization by throwing in an acid that won't, a weak acid that won't activate the polymer on its own, but might actually accelerate the depolymerization once you have an active chain end?
So the next question comes from Grace. She asks, could you add some of the polymer black containing polymer with non-carbon black containing polymer and still see degradation?
You know, that is not something we tried. I think you're basically taking a polymer sample that has carbon black in it and one that doesn't, mixing them together. So I think the net effect would be reducing, I guess it depends how well they mix together, but I think you would still see depolymerization, but this might be a strategy we can use for the mixed stream depolymerization. Since we can actually make nice films of those because the polymers don't phase mix, we might be able to just mix them together in solid phase and see depolymerization based on degradation temperature.
So Grace had a follow-up question to that, which was, are there non-solution phases back into your films?
Oh, sorry, you broke up at the end of that.
Oh, are there non-solution phase methods to get the carbon black into your films?
I think so. I think we can, I mean, the trick is not starting to break down the polymer through any mechanical grinding and processes like that. Polyaldehyde itself is a little bit harder to work with because you can't actually measure the TG of unsubstituted polyaldehyde because it's above the degradation temperature, and so you can't melt it. But if we put in plasticizer and put in carbon black, we might be able to lower the effective TG enough that we can incorporate the carbon black that way. But ideally, if you could melt the polymer and incorporate the particles, that might be a good way to do it.
I'm going to ask one more question. So do you envision a depolymerization system where you can use plasmonic energy in tandem with photothermal energy for depolymerization?
I think that's a really cool idea. So right now, we are just using thermal energy. Carbon black itself is not plasmonic. We don't think it's doing any weird electron transfer, energy transfer, or anything like that. It's just generating heat to induce depolymerization. As you get to some of these higher degradation temperatures, or if we want to help get over the kinetic barrier of depolymerization, we might need to use additional methods of activation. So using other forms of catalysis in concert with the heat to push the depolymerization. And so then using electron transfer might be really beneficial for energy transfer.
So it doesn't look like we have any more questions. So thank you, Erin.
All right, cool. Thank you all for listening.
Great. Thank you so much for two wonderful talks today. I hope that you'll join us next week again. We're almost done with this series, so I really appreciate everybody still tuning in. I know your semesters are starting, so I hope classes or teaching goes well, and I'll see you again next week. And thanks again for your patience today. Have a good afternoon, everyone.